Microplastics are found in all kinds of environments all over the world, and there's increasing concern that they may be harmful to many different kinds of organisms. Currently, it's very difficult to test for microplastics and it's very expensive to measure them. In my research group, we've developed some new methodology that allows you to measure microplastics much more quickly and cheaply than you can using conventional methodology. At UEA, we developed a technique that allows you to carefully dissolve away all of the papery content of the tea bag, and this reveals the skeleton inside of the tea bag, which is made out of plastic.
At the Quadram Institute, we are very well equipped with a variety of modern instruments for counting and sorting very small particles. The flow cytometer here is being used to count thousands of particles, identifying them based on their shape, their size and their light absorption properties. This allows us to distinguish microplastics particles from the huge background of other materials present in the sample. Once sorted, we can apply a range of other techniques to the microplastics particles, such as the specialist forms of microscopy in Andrew's group at UEA. This allows us to identify individual plastic types. In our collaboration, we want to develop a standard pipeline for quickly and reproducibly measuring microplastics in the food chain to give us some solid data from which to assess the potential risk to human health.
The work that I'm involved with Andrew on has developed over the past few years and encompasses broadly litter and microplastics research, but sometimes we discuss how to find the correct evidence that we need to answer policy questions for government. Sometimes we discuss how to do various projects in overseas countries. And sometimes we discuss the newest and best ways to actually analyse for the different microplastics and nanoplastics that are of interest.
A perfect example of collaboration between UEA and CIFUS is our collaboration for the development of a fluorescence tagging of polymers using NALRED, a technique for the large-scale mapping of microplastic in environmental samples. So this technique has been applied in many ways within CIFUS, especially through our national and international projects. So we did use this technique for the monitoring of microplastic in environmental sample for the Pacific, the Caribbean, as well as for South Africa. And we are expanding our basically scope of work with the addition of Sri Lanka, India, and the Maldives. So you can see this collaboration is very valuable for us because actually it is included in many of the work that we are doing within CIFUS.
Welcome to Newmarket Water Recycling Centre, where Anglian Water has been working with the University of East Anglia and Mott MacDonald on a piece of research to understand the macroplastics that are coming into the treatment works.
So what we have here is a rag skip full of material that's come into the wastewater treatment works from the catchment, and we have lots of interesting stuff in it. As you can see, it's not just full of organic material, it's full of plastic, it's full of wet wipes. You can see they're a couple of feet tall in height. It's got plastic toys, it's got fragmented material from hard hats, it's got wastewater, single-use plastics, it's got a load of measure of different types of plastic.
The research undertaken by UEA has been really important in better understanding the plastic item types that we receive into the treatment works, as well as a quantification of these and a better understanding of some of the polymer types that make up items such as wet wipes. This is really important for the Anglian Water and the water industry more broadly, because it allows us to extrapolate these data across all of our treatment works so we know what we're receiving through the sewers.
We're working as part of the Minimise Consortium, funded by the Natural Environment Research Council. In this project, we're using our expertise in polymer synthesis and processing to make fluorescently labelled microplastics with controlled shapes and sizes. These are used by other partners in the consortium to study the behaviour of microplastics in organisms and evaluate their possible ecological threats to the UK shelf seas. With funding from a Global Challenges Translation Award, we were able to build microplastics analysis kits based on the approaches pioneered in my group and ship them to collaborating partners. These groups have set up microplastics analysis facilities and built a rapidly growing network of collaborators, whom they are helping to train and equip through sharing of the analysis kits. We're hoping that the lessons learned from this project will help us to further expand this work to other countries where plastic pollution is really serious, helping them to assess pollution, understand environmental impacts, and ultimately to support education and policy change to reduce plastic leakage into the environment.